All right, so you will not find this one in the notes package. Okay, this is AP material. Right, so we're going over this. Okay, it could come up like, not so much like on a quiz or an exam, but it could come up when I use examples in class. I might refer back to this stuff, but it's again not something that's going to be tested. This is the AP material only. All right, so we need to be able to identify a virus from structural descriptions, know the parts of a virus. That's pretty easy. It's not like a cell because it isn't a cell. It's not made up of cells. There's very little to a virus. There's like 2 parts and that's it. Okay, understand how viruses live and reproduce, and I'm going to say we're using the word live very, very loosely here. Okay, because a virus, by a lot of definitions, is not technically alive. Okay, it doesn't do any of those things we talked about that are indicative of a living organism. Carries out no metabolism. Okay, produces no waste products. Takes in no material. Okay, can't reproduce on its own. Right, so like unlike cells where they divide to reproduce, a virus cannot do that. Okay, we'll talk about how a cell or how a virus uses a cell to help it reproduce and things like that. So by the very kind of technical meanings of life, a virus is not a living thing. Right. That said, they're quite a serious health concern. Okay? Understand why viruses are being studied so carefully by modern medical science, and identify how a virus can spread or become more virulent. What does virulent mean? Yeah, more easily contagious. Okay, also more harmful. Okay, like the symptoms may become more serious. Okay, so viral structure. First off, a virus is not a cell. Okay, they're not made up of cells. There's no membrane, there's no wall, nothing like that, no nucleus, nothing. Okay, there is debate as to whether or not they can even be considered to be a living thing. Viruses are basically an infectious particle that consists of a short string of genetic information. Some viruses have RNA only, so a single strand, and some have DNA. Okay, it's double strand, but it depends on the type of virus we're talking about. And that DNA or RNA is enclosed in a protein shell called a capsid. That's it. That's all there is to a virus. Capsid, protein shell, and the genetic material inside. That's all there is to them. They are very, very simple. And as a result, very, very small. Okay, that's what makes them so difficult for your body to destroy. Okay, because if you have a bacterial infection, the culprits are larger and easier to find. Okay, they emit toxic materials. That's what makes you sick if they're a bacteria, and so they're easy to find in your body and kill. Okay, not the case for viruses. Viruses are very small. They hide, okay, and they don't give off anything to allow them to be tracked, really. Okay. All right, so these are some of the different kind of shapes. We've got the rod-shaped stuff. This is tobacco mosaic virus. It actually doesn't infect people, okay, but it can affect tobacco plants, and it makes their leaves all mottled and like soft, so they're not good for drying and then being used for smoking. Okay, we have adenoviruses. A lot of those, okay, are kind of simple things like not the common cold, but some other diseases can be caused by adenoviruses. Okay, the influenza virus, that's the flu. Okay, what makes the influenza very tricky is that it's a capsid that's surrounded by a membranous coat. Okay, so it actually hides the proteins, and that makes it harder, even harder for your body to identify it, right? And then there are what we call bacteriophages. Okay, they kind of look like the lunar lander. Right? If you've ever seen what that thing looked like, that's kind of what they look like, and they attach to cells. Okay. So those are basically the 4 shapes that you would see. But as you can see, they're all quite small. The tobacco mosaic virus is about 10 nanometers across. Okay, this one's about 50 well, yeah, a little more than 50 nanometers. This one's about 35, 40 nanometers. Okay, the bacteriophages are probably the biggest. Right. Okay, so the protein shell that encloses the viral genome. Genome means your genetic material. Okay, your genome is inside of every nucleus of your cell. Okay, it's all of your genetic material. Okay, that's called a capsid. Okay, it comes in a variety of shapes, from simple rods and spheres to cubes or even more complex forms. Okay, the HIV virus is an incredibly complex form. It's got a capsid inside, then it's got a lipid membrane. Okay. It's a retrovirus, so it works a little bit differently. Okay, the herpes simplex virus, one that causes cold sores and things like that. Okay, it's got the capsid again inside with glycoproteins around the outside. Okay, this is the tobacco mosaic virus here on the right. Okay, oddly enough, they kind of look like cigarettes. Okay. Now, some viruses have these accessory structures. That's the the glycoproteins and the membranes that essentially cloak them, okay, from your immune system. The only way your immune system finds a virus is by the protein shell. Okay? It can recognize that that protein is not something present in your body normally and will treat it as a foreign body and attack it. The problem is viruses are very small, and when you're sick, very, very numerous. And it is very hard for your body to get rid of them all. Okay? On the plus side, a virus does not live very long outside of a cell. Okay, if you were to like sneeze on a desk, okay, because you had the flu, the viruses in there would likely be dead in minutes. Okay, they must infect a host cell very quickly before their protein shell breaks down. Remember, they can't carry out any metabolism. Okay, they can't convert energy, any of that kind of stuff, so they die very fast. So when you're looking at like cleaners and stuff, and like I think it's Lysol that says kills the flu virus in seconds, the flu virus would die in 5 minutes anyway. Okay, all it's doing is speeding up the process a little bit. So, you know, if you're getting that fresh mucus, you could kill it quick. Okay. All right. So bacterial viruses, that's the bacteriophage there. Okay, like I said, it looks kind of like the lunar lander, right? So there's these accessory structures called tail fibers that allow it to grip onto bacteria. This kind of virus only infects bacterial cells, right? So believe it or not, things that can make you sick can be made sick by other things. Weird, but true. Okay, this is actually the type of virus that's being studied by modern medicine in terms of genetic therapy. Okay, what they're trying to do is create viruses that'll have different genomes of their own choosing. Like we essentially develop the DNA that goes inside. Okay, and then they inject an animal or a person with that virus, and that virus infects the person and then changes their DNA. Okay, so it's a genetic therapy, okay, that's supposed to treat certain diseases. It's in its very early stages in infancy, but it's quite promising. Okay. All right, so viruses are intracellular parasites. That means they parasitize your cells from the inside, right? So what happens typically is a virus will attach to the cell membrane and it will inject its genetic material into the cell. Now, your cell knows what genetic material looks like. And where does it assume it's supposed to be? In the nucleus. And what's the only thing that can get in and out of the nucleus? Genetic material. Okay, so this is how viruses fool your cell. They inject their own genetic material, which is then conveniently taken to the nucleus by your own cell's machinery, okay, allowed in, and then it splices itself into your DNA. So it cuts out a section of your DNA and splices itself in between. All right, now, that DNA that came from the virus codes for one thing: how to make a virus. In the same way that your DNA is a code for how to make you. All right, so all it does is splices itself into a spot in your DNA, and the next time your cell wants the instructions that are supposed to be there, instead of making whatever's supposed to be there, it makes a virus. Okay, and here's the problem with that. The cell didn't make what it wanted. So whatever part of the cell was calling for whatever was supposed to be made calls for it again and again and again. And it's this feedback loop that starts to happen. Hey, we keep asking for this. What the hell's going on in there? Okay, we're supposed to be making this stuff and we're not getting it. What's going on? Oh, we'll make it. And they think they're making it and they're not. And eventually the cell fills up with viruses until it pops. Okay, so you can have one virus infect the cell and replicate itself 1,000-fold. So one cell can produce 1,000 viruses, okay, before it explodes, all right, or just ruptures, whatever. Okay, but that's pretty serious. That's why sometimes, you know, you go to bed and you got like the tickle in your throat, right? And you're like, oh, I think I'm getting sick, and in the morning you think you'll have to get better to die. Okay, it can happen that fast. You think about it, right? One virus produces 1,000 copies. Each one of those 1,000 copies goes out. Now, they're probably not all going to be successful, but even if only 10% of them are successful, okay, you have 100 more cases of 1,000 viruses. Okay, really can quickly get out of hand. All right, we're going to leave it there because we got to take you down to the ELL thing here. All right, so as we were saying here, okay, what goes on with a virus is that it attaches to the outside of a cell. It injects its DNA inside, and then that DNA finds its way to the nucleus, where it writes itself in. It splices itself in, okay, to the virus or to the cell's DNA, and the cell will begin to produce viruses. Effectively, it hijacks the cell. Okay, it takes it over and it makes it do what it wants, which is unfortunately produce lots more viruses. Okay, so that's kind of the life cycle there that we just talked about. There's the attachment, the injection. Okay, there's the kind of dissolution of the DNA, kind of the splicing in, the assembly and construction of viruses. Oh, Kira, can you just let them in there? Okay, and then the cell will be filled with 100 to 200, depending on the cell. It could be even 1,000 viruses. Okay, and it pops and out they come, and then they go off and infect other cells. Now, again, if they're not able to infect a cell within a few minutes, the virus just breaks down and it's dead. Okay, but if they do happen to come into contact with a cell during that short period of time, which is pretty likely because you're made of cells and they're everywhere, then they will attach and cause the infection to spread. Okay. All right, so like I said, to infect the cell, it latches on, injects its DNA, okay, and then effectively splices itself in, okay, to the cell's DNA. If the cell divides while that viral DNA is in there, guess what gets copied? The viral DNA. Then both daughter cells will have that viral DNA. They will both still be infected, okay, and it's like a generational pass of the virus from one cell to another. Okay, so if we're looking at like the HIV kind of life cycle here, okay, we got like the AIDS virus or HIV virus consists of 2 strands of RNA and some enzymes. Okay, so it will effectively attach onto a white blood cell. Okay, the virus encounters a T cell, which is part of our immune system. Proteins on the virus coating bind to receptors that are on your cell membrane. Okay, the virus enters the cell. The RNA is converted into double-stranded DNA by an enzyme. Okay, and then the third stage here, that was the 4th stage, enzyme called integrase. Okay, that's how it splices its DNA into the cell's DNA. The viral DNA uses the cell's manufacturing process, directing it to churn out viral RNA and proteins. Okay, the thing with HIV is it doesn't outright kill the immune cell quickly. It can actually hijack and use that immune cell for a very long time. What's kind of worst about HIV is that as it's detected in the blood, what does the body send out to get it? More of these cells. Okay, so it gives them more, essentially not food, but it's kind of like that. Okay, the HIV virus infects the cells that are sent to kill it. Okay, which makes it especially kind of insidious. All right. Then for stage 6 here, protease enzymes cut the viral proteins. Essentially, they make the proteins come together, okay, and form the final product, and then they bud out. Rather than rupturing the cell, they actually just bud out through the membrane. So the cell doesn't die. It keeps producing these things. All right, does that sort of make sense on how that works? Okay. Okay, so we've talked about that there, the viral life cycle. This is actually an HIV-infected white blood cell under an electron microscope. All right, the little gray pieces here that you can see are budding HIV viruses, okay, budding out of the cell. So you can see there's a number of them on that single cell, okay, getting ready to come out. Okay, so 3 processes can contribute to the emergence of a viral disease, that is for it to reappear or appear for the first time. Okay, an existing virus can evolve and cause disease in individuals who had developed an immunity to an ancestral form of it. Right, this is why you have to get a flu shot every year. Right, because every year we kind of have to take our best guess at what the flu virus is going to be for that particular year. Right, we don't always know. Okay, we kind of look at the ones that have been detected and we take our chances on that. But every year the flu virus mutates, and so there's always new versions of it. Okay, so that's one of them. Okay, or maybe you got the chicken pox as a kid. Okay, and there's a mutation in the chickenpox virus. Most of the time you only get chickenpox once. Okay, but if there's a mutation in the virus, then your immune system may not recognize it and you can get it again. All right, second one, an existing virus can spread from one host species to another. So maybe a virus that only infected your dog, okay, is suddenly mutated slightly, and now your dog gives you a big sloppy kiss, and now you're sick. Okay, it's pretty rare that that happens, but it can move from one organism to another. Okay, and then thirdly, an existing virus can disseminate from a small population to become more widespread. This is what the World Health Organization is always worried about. Okay, when that SARS virus was going around there about 8 or 9 years ago now, okay, they were very concerned because if someone was infected with it and then got on a plane, okay, they could take it from one place to another and cause what we call a pandemic, which would be a worldwide infection, okay, of that particular virus. Okay, Ebola is another one. Okay, it's like if you come in from a country that has had Ebola cases and you start to feel sick and you go to the hospital and they ask you, were you in any of these countries? They will quarantine you immediately. Okay, because you could be infected with that and they don't want that to get into another population, a larger population or something like that. So that's a big concern. Okay, that was also the case with HIV. Okay, for the longest time, HIV was really only known to be in the homosexual community. Okay, when it was first discovered, that's where the bulk of the cases were found. But then obviously it disseminated from that population into also the heterosexual community, and now of course it's kind of in both. All right, but it was generally found to only be in one. It was probably in the heterosexual one as well, but it wasn't as common. It was mostly diagnosed early on, okay, in just one community, but then disseminated from there. Okay, and that could have been the result of contaminated blood. There were a number of cases of people that got HIV because we didn't know about it. We didn't know it was in blood. People donated blood and they had it, and people received blood that was infected. Okay, obviously now they test for that. Okay, before they give blood, so it's not something you have to like go home and stress about. Okay, that won't happen now. Okay, all right. So that's all we need to know about viruses. Okay, we'll cut it off there.